Hi friends, in this video let us understand what is microbiome engineering, what are the steps or basic procedures involved, and also its application within 5 to 10 minutes. Let's begin with what is microbiome engineering. Microbiome engineering is the purposeful manipulation or modification of microbial communities through the addition, removal, or modification of specific microbes, genes, or community structures to confer beneficial functions to hosts such as humans or plants. Let me make it more clear with an example. Our human gut is home to trillions of bacteria and other microorganisms that is essential for our survival. Any change in this normal flora can lead to many chronic human ailments like obesity, type 2 diabetes, non-alcoholic fatty liver, etc. So we will be restoring this microbiome through microbiome engineering for restoring our health. Let us understand with an example of microbiome engineering process. Let's take gut microbiome engineering as an example. It is one of the most studied microbiome. Step 1 is define engineering objective. That means identify the specific biological function or therapeutic goal to be achieved by microbiome engineering. Let us understand this with an example. Suppose we want to alleviate IBD or inflammatory bowel disease through secretion of anti-inflammatory cytokines by engineered Lactococcus lattice. E. coli and Lactococcus lattice are the common model gut bacteria isolated for engineering. Step 2 is characterize native microbiome. We need to study about the native microbiome using multiomics techniques, metabolomics, transcriptomics, metagenomics, etc. to analyze the composition and function of the target microbiome ecosystem. We should have a thorough understanding of the normal flora or normal microbial flora of gut microbiome. For more details, you can watch our video on metabolomics. Step 3 is isolate and select target microbes. So here we need to culture or identify native gut bacterial strains. Here we are using Lactococcus, relevant to the engineering call for modification. So we'll be isolating Lactococcus from the gut microbiome. Step 4 is genetically engineer this Lactococcus colonies. There are different techniques available for gene editing like CRISPR-Cas9, conjugation, electroporation, etc. to add, delete, or modify genes that confer desired functions such as therapeutic molecule production or metabolic pathway alteration. Here our intention is to genetically engineer a Lactococcus by addition of genes for producing anti-inflammatory cytokines that is in tune with our objective. The next step is design synthetic microbial consortia, and this is an optional step. If we are working with multiple strains, this assemble multiple engineered strains to create a community which will be having a synergistic or additive effect. In this case, we are using a single strain, so this step is not essential. Next step is ensure colonization and stability. Here, we need to optimize delivery methods like providing selective nutrients to promote persistence of engineered microbes in the gut environment. And this is the most difficult step. The engineered microbiome should thrive well in the gut environment. So we will be devising different methods like providing specific nutrients so that they can grow optimally under the gut condition. And finally, test and optimize in model systems. We'll be using model systems to evaluate the engineered microbiome's function, its stability, and host interaction using animal models or gut-on-chip systems to find out whether there is any side effects or any change in metabolism, what are the beneficial aspects, and what are the things that can be improved. And based on this, we improve designs or we optimize specifically that suit our purpose effectively. And finally, the clinical trials or clinical application. Then we'll be conducting safety and efficacy trials with engineered microbes in humans or target hosts with continuous monitoring of microbiome and health outcomes. And these are the basic steps or procedures that is usually followed in microbiome engineering. Now let us see the application of gut microbiome engineering Gut microbiome engineering is widely used for treating metabolic disorders. In the case of phenylketonuria, we are using engineered bacteria metabolizing pathological compounds. Then it is used for alleviating IBD or inflammatory bowel diseases through secretion of anti-inflammatory cytokines engineered using Lactococcus lactis. We have already discussed that. Then modulating bile acid profiles to influence host metabolism and immunity. Then engineering gut microbes to improve gastrointestinal motility via metabolic product synthesis. For example, tryptamine production from tryptophan amino acid. Then we will be providing novel biotherapeutics or live microbes with controllable colonization for chronic disease management like obesity, type 2 diabetes, etc. Now let us see the applications of microbiome engineering in general. We have already discussed it is widely used for treating metabolic disorders, especially human health. We use engineered gut microbiome to improve digestion, treat metabolic and disorders and inflammatory diseases, and support drug metabolism. In plants, it is widely used to enhance agricultural productivity by enhancing plant growth, stress resilience, nutrient cycling, and disease resistance through modification of root and leaf microbiomes. Other applications include food safety and quality, modulating microbial communities during food production to prevent spoilage and pathogenic contamination. Then it is used in environmental management, restoring ecosystem functions, bioremediation of pollutants, and finally, it is used in biocontrol and sustainability, reducing the need of chemical pesticides and fertilizers by shifting microbiome composition towards beneficial, naturally occurring microbes. So these are some of the applications and some of the areas where research is going on with regard to microbiome engineering. Hope you are clear. And these are some of the references. Thank you so much. You are with biologyexamsforyou.com.